Assalamu alaikum everyone, this is RBHV and welcome to the platform of Pharmaphilia once again with a new topic of blood physiology. This is basically the interpretation of CBC series. So let's begin the video. This is the first video of this series. Blood is a special type of the fluid connective tissue derived from the mesoderm, the branch of the spine concerned with the study of blood and the blood forming tissues. That means the tissues that form the bloods and the disorder associated with them is called the hematology. Hem is blood, this is obviously blood, and the logos means study. Now the properties of the blood, as you can know, its color is bright red in arteries and dark red in veins, because in arteries it contains the oxygenated blood and in the veins it contains the deoxygenated blood. Mass 8% of the body mass and its pH is slightly alkaline, that is pH 7.35 to 7.45. Its taste is salty. Temperature 38 degrees centigrade is the normal temperature of the blood, and the viscosity 3 to 4 times more viscous than water. Now its volume 5 to 6 liter in the body. Composition of blood. Blood is composed of white blood cell, platelets, and red blood cell. As we learned in the primary school, blood is composed of these three components, but it is also composed of plasma. As you can see, the upper yellowish part is known as plasma, and the lower red part is called as red blood cell. And this picture you can see when we centrifuge the blood in the lab. Now, how the RBCs and how the other blood cells are formed. These are basically derived from the multipotential hematopoietic stem cells, and it further divided into the common myeloid and lymphoid progenitor cells. These common myeloid progenitor cells is further divided into the megakaryocyte, and which is converted into the thrombocytes. Thrombocyte is the another name of platelets. From the common myeloid progenitor cells, erythrocyte, that means RBCs, are formed. Mast cells, as you can know, mast cells release histamine that have the major role in the allergy reactions. Myeloblast further divided into the basophils, neutrophils, eosinophils, and monocyte. These are basically the type of WBCs. Now, when we talk about the lymphoid progenitor cells, it is further divided into the natural killer cell and the small lymphocyte. Lymphocytes have role in the inflammatory cascade, so it is further divided into the T lymphocyte and B lymphocyte. Another type of the cell produced from the lymphoid progenitors are natural killer cells. Hematopoietic stem cells present in the red bone marrow, and it further form the red blood cells, white blood cell, and the platelets, as you can see in the picture previously. Now, what are the components of the blood? It composed of the plasma and cellular component. Cellular component is further divided into the RBC, WBC, and platelets. Plasma is basically present in the major proportion, that is 55%. Now, what is a plasma? Plasma is a pale yellow colored liquid component of the blood. This pale yellow colored liquid we get when we centrifuge the blood in the hematology lab. Okay, that hold the cellular elements of the blood in suspension, and constituents of the plasma are water, that is a major proportion of the plasma, 91.5%, protein, that is 7% protein present in the plasma are albumin, globulin, and fibrinogen, and the other solutes present in the plasma are electrolytes, iron, gases, nutrients, and the waste products. Waste products are urea, uric acid, ammonia, and the creatinine. Now, the function of the plasma. It constitutes of water. What is a function of the water? It absorbs, transport, and release heat. What does it mean? It means it helps to maintain the dynamically and thermodynamically stability in the body. Now, albumins help in balancing the osmotic pressure of the body. Globulins have the major role in the defense of the body. What is the other type of the cell which have the same functions? That is the WBC, which is called the police of the body. Fibrinogen help in blood clotting and electrolytes ion help in pH buffering. Now the red blood cells having the biconcave non-nucleated circular in shape with a diameter of 7 to 8 micrometer and 2.5 micrometer in thickness. Color is red because of its hemoglobin pigment. Now its count in adult is 5.4 million RBC per microliter and in adult female 4.8 million RBC per microliter, and its lifespan is 120 days. We will discuss in detail the basics of the RBCs and the disorder caused by the elevation of RBC count. Now, erythropoiesis. What is a erythropoiesis? The production of the RBC is known as the erythropoiesis. Increase in the number of RBC is known as the polycythemia. And what is a major cause of the polycythemia? That is smoking and the alcohol consumption and any secondary disorder that is called the secondary causes, which is the cancer, leukemia and other chronic disorder, etc. Now, decrease in the number of RBC is known as the erythropenia. In adults, red bone marrow of long bones, that is a hip bone, breastbone and the ribs, these are basically the site of the erythropoiesis, and in children up to the 5-year bone marrow of all the bones, and the fetus, liver and spleen. Now, function of the RBC, we can learn in the primary school that the basic function of the RBC are transport oxygen from the lungs to the tissue and transport carbon dioxide from the tissue to the lungs. Normal blood contains 13 to 50 gram of Hb per 100 ml of the blood, and one RBC contains about 250 million molecules of Hb. Each molecule of Hb carries the 4 molecules of oxygen because it transports oxygen from the lungs to the tissue. White blood cell, another type of the cellular element of the blood. Its shape is amoeboid nucleus. As you can see, it is amoeba-like in the picture. Now its size is 12 to 15 micrometer and it is colorless and translucent. Its normal count is 5,000 to 10,000 WBC per microliter and lifespan is 10 to 13 days. And what is the lifespan of the RBC or the normal RBC? That is 120 days. Now, leucopoiesis. Leucopoiesis is the production of the WBC. Leuco means WBC. Increase in the number of WBC is known as a cytosis. So what is the meaning of cytosis? When you can see anywhere this cytosis, that means there is an elevation or the increase in the cell count. And when you can see the word mania, that means there is decrease in any cellular count. So the decrease in the number of the WBC is known as leukopenia, and the pathological increase in number of WBC is known as leukemia. Leukemia is another name of the blood cancer. In other, liver, spleen, tonsils and bone marrow is a basis site of leucopoiesis, while in the fetus, as same for the RBCs, that is the liver and spleen. Type of WBCs. WBCs are further divided into the two types on the basis of its granules. Granular WBCs are neutrophil, acidophils and basophils, and agranular are lymphocyte and monocytes. Now the features. Neutrophils is having the nucleus with 3 to 4 lobes, as you can see in the picture, 1, 2, 3 and 4 lobes, and its basic function to destroy the bacteria by the phagocytosis. Its location of production is bone marrow. All of the WBCs are produced in the bone marrow, so this is a similarity in all of the type of WBCs. Now acidophils, which is commonly known as eosinophils. Eosinophils is the commonly used term, and you can see this term in the CBC when we discuss the CBC in the next video. Its featured nucleus with the two lobes. As you can see, this is a headphone-like structure in its appearance, stained with the acidic dye, and its function compact the function of histamine in the allergy reactions. Basophils, nucleus with the indistinct lobes, stained with the basic dye, liberate heparin and histamine in the allergy reaction to intensify the inflammatory response. What are the agranular WBCs? Agranular WBCs are lymphocytes and monocytes. Features smallest of the WBC is the lymphocyte and large round nucleus. It produces the antibodies. Additionally, with the bone marrow, its location of production is spleen. Also with the spleen, tonsils. Monocytes, largest WBC, and the smallest WBC is what? Lymphocyte, and the largest WBC is monocyte. Large kidney shaped nucleus. Here you can see the purple color structure, kidney shaped nucleus in just microorganisms. Platelets are biconvex, non-nucleated in shape with a size of 2 to 4 micrometer, and its count in 1,50,000 to the 4 lakh platelet per microliter. This is a huge amount in the body, but this is the normal range also. Remember this. Lifespan is 5 to 9 days, and its function, it helps in the blood clotting. Thrombopoiesis, the production of the platelet is known as the thrombopoiesis. Platelets are the fragment of the large cells called the megakaryocyte that remain in the bone marrow. Increase in the number of platelets is known as thrombocytosis, and decrease in the number of platelets is penia. Penia of what? Thrombocytes, so it is called the thrombocytopenia. Blood clotting. Blood clotting is the process in which the blood loses its fluidity and become a jelly-like mass few minutes after it is shed out. And the process of the blood clotting: first, blood vessel is punctured and platelet form the plug. Platelet are damaged. Tissue cell release prothrombin activator. Prothrombin is basically the clotting factor which initiate a cascade of the enzymatic reaction. So fibrin thread, as you can see in the picture, where the blood vessel is punctured, here the fibrin thread is formed and trap the RBCs. Now anticoagulant, a substance which prevent the coagulation of the blood, is known as anticoagulant. Heparin is a natural anticoagulant present in the blood. If blood clot too easily, the result can be thrombosis, clotting in an undamaged blood vessel. If the blood take too long to clot, is called the hemorrhage. That means the excessive blood loss. And that's all from this topic. That is a basic physiology of the blood. This is the first video of the interpretation of CBC. Hope you like this video and got the basic concept. Till the next video, take care. Allah Hafiz.